नमस्कार दोस्तों, वेलकम टू चैनल टॉक्सिकोलॉजी ऑन बोर्ड. Today in this video we'll be discussing about another DABT topic, that is functional observation battery with respect to pupillary response to light. So let's directly dive into the question. The question is, how in the functional observation battery would one evaluate the potential effects of a toxicant on vision? So important thing what you have to remember is FOB, part of safety pharmacology study. No, we have three different organs will be there. One is central nervous system, cardiovascular system, and respiratory system. In FOB CNS related, there are few tests, FOB tests, which are related to visual evaluation, visual inspection. So that related question will be there, that related concept will be there in this video today. So we have, once again, let me repeat the question. How in the FOB would one we evaluate the potential effects of toxicants on vision? So FOB में various parameters are there, like to understand the grip strength, to understand cognition, memory, all these things will be there. So in that, this one of the test in the case of FOB with respect to vision is also there. So we need to see. So we have option A, we have evaluation of ocular irritancy. Option B, we have observing the pupillary response to light. Option C, we have scoring the degree of lens opacity. Option D, we have evaluation of rod and cone density. So the right answer will be green text, that is option B, observing the pupillary response to light. So let's see what is the reference which is explanation and the reference which is given by the American Board of Toxicology. Only two visual responses are tested in the FOB, right? What are those two? One is pupil contraction based on the light, and the approach to the eye. So let us see what further reference are given under Cassette and all 9th edition, page number 888. So let's see the next slide. So under the Clayson textbook on page number 888, this is what information I have come across in the textbook. So you can directly read in the textbook, or you can go through this slide also. So visual toxicity screening procedures under FOB, what they have given, we'll see. As noted above, there is a clear need for retinal and visual inspection testing as a component of this toxicological evaluation of environmental and occupational chemicals. So there is a need to test the retinal as well as visual function. How we are going to do? The extent and magnitude of potential retinal and central visual system alterations in humans and experimental animals exposed to most drugs and toxicants is not known because this has not been systemically evaluated. Okay, still there is no proper methods are established to check for this particular thing, extent and magnitude of potential retinal and central visual system alterations in the humans as well as in the case of experimental animals. The EPA as well as OECD, Environmental Protection Agency, and Organization for Economic Cooperation and Development, screening batteries for neurological evaluation, neurotoxicological evaluation. As I told you, it's a part of CNS. So, neurotoxicological evaluation of laboratory animals only include minimal evaluations of this visual inspections. What are those minimal evaluations? Functional observation battery, and automated measurement of motor activity and neuropathology. So this is a reference which is given under EPA. Only the FOB evaluates the visual function. So our question was also with respect to that only. What only FOB evaluates the visual function, and extent of these measures is very limited. Okay, the entire assessment of this visual function includes observing the animal's response to an approaching object such as pencil, and observing the pupil's response to the light. So only two things are there. One is that near your eye, no, they try to bring the object. Generally, veterinarian or the study directors, what they do is, whenever they're performing this functional observation battery test, which is a part of either 90-day test and sometimes 28-day repeat dose toxicology study, what they do is that they try to bring the pen near the eye of the animal, and they will try to see the response of the animal. So that is what, see, they are saying, observing the animal's response to an approaching object such as a pencil, and observing the pupil's response to light. One other thing what they carry always, the study directors, is that they have a light, a torch will be there, and they will be putting that light into the animal eye, that is rat eye, and to see how it is, how the pupil response is. Generally it is will be this visual related test will be done by the veterinarian who is present in that particular GLP facility. Okay, so that is called Vida Supra. So let's see what further information is given under Clayson textbook. So under the continuation of this particular paragraph in the textbook on page number 888, visual toxicity screening procedures. These procedures are limited as they do not explore responsiveness over a range of stimulus features such as luminance, how the animal is reacting or responding to luminous, how it is responding to different colors, red, blue, green, like that, and how it is responding to contrast, spatial frequency and temporal frequency. So if you look into FOB, only two things they are doing. One is bringing the object here and there, and light response they are doing. All other these things they are not checking. Luminance, color, contrast, all these things are been not been part of this FOB. Next one, in addition, they do not, means whatever the in the visual inspection in under FOB, no, they do not evaluate rod or cone sensory thresholds. Since a part of the first picture you see, the part of the important eye is rods and cones are there, no? So those they are not evaluating this rod or cone sensory threshold or maximal responses, nor do they isolate potential motor or integrative contribution to task performance. So all these things are also not being evaluated under this FOB visual inspection. Despite these shortcomings, FOB has been successful at detecting visual deficits produced by exposure to, this is an example they are giving, 3,3 propionitrile or carbon disulfide. So in the ocular toxicity only, please go back and read. They have given, whenever there is a target organ toxicity in this textbook, no, at the end they will give you some 8 to 10 examples chemicals they will be giving, and they will be explaining about the toxicity. So in that list I have seen that carbon disulfide is also there. So go back and read how these chemicals are causing ocular toxicity. In the eye also different regions will be there, how the which part of the eye they are affecting, all those things you see, which illustrates the importance of conducting at least this level of visual function screening in routine testing program. Okay, some studies include ophthalmological and ocular pathological evaluations. So you go back and read once again if you look into OECD 407 guideline, when you do histopathology of 28 to 30, 32 organs you will be doing, see if eye is also a part of that particular thing. Those who are doing studies, they will be definitely doing. Other people, please go back and read and check whether the eye is a part of this one. Are they performing histopathology? That is an assignment to you people. Some studies include ophthalmological and ocular pathological evaluations. Comprehensive visual toxicity studies should include ophthalmological, histological, pathological evaluation of ocular tissues, as well as assessment of simple and complex visual function. So from this, I hope you will able to understand the importance of this visual inspection being a part of functional observation battery. Okay. So thank you so much. Be curious. For any questions, please write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. Namaskar. All the best for your exam.